Swayam Prabha. Digital India. Educated India.
Hello everyone, I am Dr. Rose, Assistant Professor, Department of Anatomy, Government Medical College, Thrissur. In this session, we will be dealing with the topic implantation and the first week of intrauterine life. So, suppose a lady comes to you, to the casualty, a pregnant lady comes to you to the casualty with history of bleeding and she is in preterm labor. This is how it will look like. And we can see that the placenta is almost covering the internal os and the bleeding is actually happening from the placenta. This condition is known as placenta previa. So, in order to understand the details of placenta previa and the clinical conditions arising from it, we need to know about the details of implantation. So, before moving on to implantation, let us see what do you mean by a gestational period. So, the normal gestational period is said to be 40 weeks or 280 days after the last menstrual period. The normal gestational period for a human being is said to be 40 weeks or 280 days, and these days we count since the last menstrual period, since the first day of the last menstrual period to be more exactly. And how will we calculate it? If we know the first day of the last menstrual period, the calculation goes like this. We add 9 months plus or minus 7 days to the first day of the last menstrual period. That is how we calculate the 40 weeks or 280 days of normal gestational period. And if we are counting the days from the period of fertilization, from the moment of fertilization, it will be roughly around 266 days or 38 weeks. So, if you are calculating the gestational period from the point of fertilization, it is just 2 weeks less. And if we calculate the gestational period from the first day of the last menstrual period, it will be 2 weeks more than the calculation which we have done from the point of fertilization. So, if this is zygote, this is after fertilization, till a baby is born, this period is roughly 266 days or 38 weeks. But if we are calculating the gestational period from the first day of the last menstrual period, that will account to 280 days or 40 weeks. This difference you should keep in mind. And in order to get this period, we have to add 9 months plus or minus 7 days to the first day of the last menstrual period. So, we have the zygote, we have the newborn, and the period in between is known as the gestational period. So, we can divide this whole period into two. The first one being embryonic period. So, this period is only up to the first 8 weeks of intrauterine period. So, the first 8 weeks we call it as embryonic period, and the rest of it from 8 weeks onwards till a baby is born, you call it as fetal period. Now, the embryonic period is further classified into two. The first 3 weeks of the 8 weeks, you call it as germinal period, and the remaining from 4 weeks to 8 weeks, you call it as embryonic period proper. So, the gestational period, the entire gestational period starting from the point of fertilization till the baby is born is divided into two, and the first half, it is not exactly the first half, it is just after 8 weeks of fertilization and beyond 8 weeks of fertilization. The two periods corresponds like that. So, the first 8 weeks we call it as the embryonic period, and after 8 weeks up to the baby is born, you call it as fetal period. The embryonic period is further classified into two. The initial 3 weeks of the embryonic period, we call it as germinal period. Why we are calling it as germinal period? Because it is during this time the 3 germ layers of the embryo are formed. So, the trilaminar germ disc, the formation of trilaminar germ disc is completed by the end of 3 weeks. So, the initial 3 weeks are known as germinal period. The rest from 4 to 8 weeks, you call it as embryonic period proper. So, the initial 8 weeks of the entire gestational period is known as the embryonic period. After 8 weeks, we call the conceptus as fetus. So, before 8 weeks, we should not call the conceptus as fetus. That point you have to keep in mind. Now, let us see the changes happening in the first week of intrauterine period. So, it mainly includes the journey of the fertilized zygote from the time of fertilization to the point of implantation. So, these are the changes which happens during the first week of intrauterine period. So, the events coming under the first week are classified as, first we have the cleavage division. You can see soon after fertilization, the cells will start dividing, and this division is known as cleavage division. Then the next step is the formation of morula, which is followed by the formation of blastocyst, and after the formation of blastocyst, it will get implanted within the uterine cavity. So, it is the journey. We have the fertilized ovum here in the ampulla of the uterine tube, and it starts its travel in order to reach the uterine cavity. So, it is actually a journey of the zygote from the point of fertilization along the uterine tube until the point where it gets implanted. That is known as the changes which are happening during the first week of intrauterine period. Now, let us see one by one. The first one is known as the cleavage division. Cleavage division, what is happening during the cleavage division? The zygote undergoes repeated mitotic division, and the zona pellucida will be still covering the zygote. That point you have to keep in mind, because till the point where you have the zona pellucida, the zygote won't get implanted. So, before implantation, the zona pellucida which is covering the zygote should be removed. So, during cleavage division, the zygote will be undergoing repeated mitotic division without causing any change to the zona pellucida. The zona pellucida, the size of the zona pellucida will remain as it is, and all the divisions will happen within that zona pellucida. That is one important point you have to keep in mind. So, what happens? The cells become smaller and smaller as they divide, and these smaller cells are known as blastomeres. So, the pattern of cleavage is like, from the zygote you have a 2-cell stage, then you have a 4-cell stage, then you will be having an 8-cell stage, then you will be having a 16-cell stage, likewise it will go. And this duration after fertilization will be around 30 hours to 96 hours, that is the estimated time period. Then what happens in the 2-cell stage? Ideally speaking, in the 2-cell stage, the cells are of unequal size, and at 12 to 16 cell stage, it is said that the cells, almost all the cells formed at this point of time will be of equal size. So, we can see the 2-cell stage, then the 4-cell, then the 8-cell, then the 16-cell. So, it is said that 12 to 16 cell stage, the cells will be almost of equal size. Now, what do you call the 12 to 16 cell stage of the dividing zygote? This stage is known as morula. Now, what happens? The zygote already has started traveling from the ampulla along the uterine tube, and roughly say after 3 days of fertilization, the zygote will be reaching the uterine cavity. Now, let us see what are the factors which help the transport of zygote from the ampulla into the uterine cavity. So, the factors facilitating the transport of zygote are, the first one, the ciliary beads of the tubal lining. You have the uterine tube, and which are having a lining, that is the ciliated epithelium, ciliated columnar epithelium, that is a lining we have for the uterine tube. So, the ciliary beads of the tubal lining will facilitate the transport of zygote towards the uterine cavity. Anything else? Yes. The second one is the contraction of the uterine tube. The contraction of the uterine tube will be acting just like the peristaltic movement along the intestine, because the peristaltic movement along the intestine will help to propel the food down. Likewise, the contraction of the uterine tube will help to propel the already formed zygote towards the uterine cavity. Now, we have many blastomeres inside the zona pellucida and they are held close together. The spherical blastomeres are held close together and this process is actually known as compaction. So, the spherical blastomeres are held in position within the zona pellucida by a process known as compaction. And the cell contact between each other, the cell contacts which you get between these blastomeres, they are mainly tight junctions. So, let us have a look. After 3 days of fertilization, the fertilization happens here. This is the ampullary region. It travels along the uterine tube by these two mechanisms. One, the ciliary beads of the tubal lining, and the other one, the contraction of the uterine tube. All these will help to propel the zygote which is fertilized in the ampulla towards the uterine cavity. So, roughly 3 days after this event, 3 days after the fertilization, this 12 to 16 cell stage, the morula, will be almost reaching towards the uterine cavity. And we can see that the cells are confined to the zona pellucida, and these spherical blastomeres are held in position by a process known as compaction, and there will be the formation of intercellular tight junctions, and these tight junctions will help to maintain all the blastomeres together. Now, the next step is the formation of morula. So, we have already mentioned that 12 to 16 cell stage is known as morula. Let us see what are the parts of morula. In the beginning, it is just a clump of cells. Later, it gets transformed into a cell mass which is seen towards the outer aspect and a cell mass which is seen towards the inner aspect. The cells which are seen towards the outer aspect, you call it as outer cell mass, and they are the cells which are seen towards the periphery and they are connected by tight junctions. So, the cells which are pushed towards the periphery and which are forming an outer cell mass are connected together by tight junctions. And what is the role of this outer cell mass? They are protective and nutritive coverings of the embryo. So, the two main functions are they protect and they also provide nutrition to the embryo. So, we have the trophoblast and the chorion, the two important structures forming from this outer cell mass. So, tropho means, the word meaning of tropho itself is nutrition. So, we have the trophoblast and the chorion developing from the outer cell mass. The next group of cells which are pushed inner to the inner aspect of the morula, you call it as inner cell mass. So, where will you get the inner cell mass? The cells of the inner cell mass will be somewhat in the center and they will be loosely arranged. They will not be tightly packed, they will be loosely arranged and there will be enough gap junctions between the cells. And what is the fate of this inner cell mass? Inner cell mass is the one which is getting converted into embryo proper. So, the parts of the embryo will be developed from the inner cell mass. At this stage, one important point you have to keep in mind is morula is still covered by zona pellucida. So, you have the morula still covered by zona pellucida. That is the reason why we are not having the morula being implanted within the uterine cavity. What is the reason? Because it is still covered by zona pellucida. In order for the implantation to take place, this zona pellucida should be removed. So, morula is still covered by zona pellucida. You have to keep that in mind. Now, we can see the outer cell mass. The cells are at the periphery and they are closely arranged with tight junctions. They are actually covering the inner cell mass. So, this acts as a protective and nutritive covering of the embryo. The cells which are grouped towards the inner aspect of the morula, which are lying in the center, this group of cells you call it as inner cell mass. When compared to the outer cell mass, these cells are loosely arranged and there will be gap junctions between the cells. And this group of cells develop into the embryo proper. That is, the parts of the embryo are developed from this inner cell mass. Still, the morula will be covered by an outer covering. That outer covering is known as zona pellucida. That is the reason why morula is not getting implanted within the uterine cavity. Now, we can see the formation of blastocyst. The morula, the cells will be undergoing mitotic activity, but at this stage the uterine secretions, otherwise we call it as uterine milk, enters the morula by diffusion. Why is it called uterine milk? It is called uterine milk because there should be some nutrition to the growing embryoblast. So, since this uterine secretions are providing nutrition to the growing embryoblast, we call it as uterine milk. So, the uterine milk will start entering into the morula through the outer cell mass. So, you might get a doubt, like we have already mentioned that the outer cell mass, the cells are arranged so close to each other with tight junctions, then how come the uterine milk enter into the morula? We can give the explanation as the secretions are entering through the cell and not through the tight junctions. The secretions are entering into the morula through the cells and not through the tight junctions. So, the uterine milk will enter into the morula by diffusion. As a result, what will happen? When you look the morula, you will be seeing many tiny fluid filled spaces or cavities which are filled with uterine milk. That is known as blastocoele. So, these fluid filled cavities will be very small in number in the beginning. Gradually they will become larger and larger, and they will be coalescing with each other to form a large cavity filled with uterine milk, and that is known as blastocoele. So, what do you mean by a blastocyst? So, blastocyst is nothing but 32 to 64 cell stage of morula with blastocoele, because till this moment we were talking about morula, which has been differentiated into an outer cell mass and an inner cell mass. In order to provide nutrition to the developing embryoblast, we need uterine secretions. So, the uterine secretions are entering into the morula in the form of uterine milk. Once they enter into the morula, the smaller cavities will coalesce to form a larger cavity. That cavity is known as blastocoele. So, previously we had a morula, now we have a blastocoele. So, morula with blastocoele all together you call it as blastocyst. So, blastocyst, the cell stage is 32 to 64 cell stage. And when is the formation of blastocyst happening? This happens roughly from the 4th or 5th day after fertilization. So, it is during this period the blastocyst is being formed. Now, what about the covering of blastocyst? Till now we mentioned about the zona pellucida covering the morula. Is it present even after the formation of blastocyst? Yes, we have the blastocyst still covered by the zona pellucida. So, this blastocyst is here, that is nothing but morula with the blastocoele. It is still covered by zona pellucida. Now, the same outer cell mass and the inner cell mass are redefined as two other terminologies. So, we have to be familiarized with these two terms, because in further development we will be calling the outer cell mass as trophoblast. So, the same outer cell mass which was lying towards the periphery is now renamed as trophoblast. So, after the formation of blastocoele, the outer cell mass we now call it as trophoblast. And what about the inner cell mass? Inner cell mass is now known as the embryoblast. And we can see that because of the formation of this large cavity, the embryoblast is actually pushed towards the periphery. Previously, the embryoblast was lying in the center, but due to the formation of this large cavity, the cavity is known as blastocoele, the embryoblast is pushed towards the periphery. So it is just a renaming. The outer cell mass is known as the trophoblast and the inner cell mass is known as the embryoblast. Why we called the outer cell mass as the trophoblast? Outer cell mass is the layer which is giving rise to the protective covering and the nutritive function of the embryoblast. So that is actually giving rise to the formation of placenta. Because of that reason, the outer cell mass is known as trophoblast. What about the inner cell mass? Inner cell mass is actually giving rise to different parts of the embryo. Hence, you call it as embryoblast. Now, depending upon the attachment of the embryoblast, we can see that the embryoblast is actually getting positioned towards the periphery because of the large blastocyst. So, depending upon the attachment of the embryoblast, the trophoblast can be again renamed. How we are going to rename it? The first one is polar trophoblast. So, polar trophoblast is the trophoblast to which the embryoblast is attached. So, you can see that this outer covering is the trophoblast, but only this region is actually getting the attachment of the embryoblast. So, this part is known as polar trophoblast and this pole you can call it as the embryonic pole. So, embryonic pole is the pole to which the embryoblast is attached and the part of the trophoblast lying closer to the embryoblast you call it as polar trophoblast. Now, what do you mean by mural trophoblast? So, mural trophoblast is the covering of the rest of the blastocyst, that is the trophoblast apart from this polar trophoblast which is forming the outermost cell mass. This is known as the mural trophoblast. This will be covering the rest of the wall and the embryonic pole, because embryonic pole is the pole to which the embryoblast is attached. So, the rest of another pole which is lying exactly opposite to it, you call it as embryonic pole. Now, let us see some of the functions of the zona pellucida, because since fertilization we have been talking a lot about zona pellucida. Even during cleavage division you have zona pellucida, even during the formation of morula you have zona pellucida, even during the formation of blastocyst you have zona pellucida. So, what is the role of zona pellucida? The first one is it acts like a barrier for the sperms, that is it allows only sperms of the same species to enter the egg. So, in human beings the zona pellucida will allow the sperm from a human being alone to enter into the oocyte. So, that is the first function of zona pellucida. It acts like a barrier allowing only the sperms of the same species to enter the egg. What is the next function? It initiates the acrosomal reaction. So, when the sperms reach the zona pellucida, it will induce the acrosome reaction. So, acrosomal reaction won't happen for all the sperms, but it is meant for the sperm which is destined to enter into the secondary oocyte. So, it is the zona pellucida which initiates the acrosomal reaction. This is how the acrosomal reaction begins. The third function of zona pellucida is it prevents additional spermatozoa from reaching the zygote. That means once a sperm enters into the secondary oocyte, this zona pellucida will act like a shield. That is, it won't allow any further entry of the sperm into the secondary oocyte. That is the main function. Again, that is one of the main functions of the zona pellucida. It prevents the additional spermatozoa from reaching the fertilized zygote. The next one is the uterine secretions reach the embryo through zona pellucida, because the placenta, the formation is not complete. So, till then the growing embryo needs nutrition. That is possible only with the help of uterine secretions which is entering into the developing embryo in the form of uterine milk. So, zona pellucida should give a way for the uterine secretions to reach the embryo. So, the uterine secretions or the uterine milk will be reaching the embryo again through zona pellucida. The next function is the zona pellucida lacks histocompatibility antigens. So, this will be acting like an immunological barrier between the mother and the embryo. Why is it called as an immunological barrier? What will happen if some foreign body enters our body? The body will try to expel it. Likewise, the newly formed embryo is actually a foreign body as far as the mother is concerned, but it is actually hidden under the zona pellucida. That is the reason why zona pellucida is considered as an immunological barrier. Then it holds the blastomeres together. So, the multiplication of the blastomeres happen within the zona pellucida. So, as and when the new cells are formed, these cells should not get dispersed. So, that is prevented. That is, the blastomeres are held together by the zona pellucida. It also helps in the differentiation of the trophoblastic cells. Another important function of zona pellucida is it prevents the premature implantation. What do you mean by premature implantation? We have already said that the morula is covered by zona pellucida, the blastocyst is covered by zona pellucida, and only when the zona pellucida is removed from the blastocyst, it will be ready for implantation. So, suppose if the zona pellucida is actually shed off from the morula stage, what will happen? It will get implanted within the uterine cavity. So, it is the zona pellucida which prevents the early implantation of the dividing zygote. If there is no zona pellucida for the morula, it will get implanted. It is just like we are just peeling off the sticker. The zona pellucida acts like a sticker in this case, because as and when we remove the zona pellucida, it will just stick onto the uterine cavity. So, zona pellucida actually prevents the premature implantation. Now, it is time for the implantation. At the time of implantation, the uterus will be in the secretory phase. So, that is the second phase, secretory phase. And during this phase, we know the endometrium has got three distinct layers. Which are the three distinct layers? The stratum compactum, which is the innermost. Then you have the stratum spongiosum. Then you have the stratum basale. The stratum compactum and stratum spongiosum together known as stratum functionalis or stratum functionale, because this is the functional layer which usually sheds off during menstruation, and the next layer is known as stratum basale. So, this is the stratum basale and this is the stratum functionale, and we have already said that the stratum functionalis is made up of stratum compactum and stratum spongiosum. Roughly say about by the end of 5th day after fertilization, it is time for the zona pellucida to move off. So, zona pellucida disappears by 5th day of fertilization. What will happen if zona pellucida moves off the blastocyst? Now, it is time for the blastocyst to get attached into the uterine cavity. So, the blastocyst will move towards the uterine cavity, and it is the implantation of polar trophoblast. Polar trophoblast is the point at which the embryoblast is attached to the trophoblast. This entire thing is called the trophoblast, and to the point at which the embryoblast is getting attached to the trophoblast, this portion is known as polar trophoblast. So, it is this point which gets attached to the endometrial wall first. So, the implantation of polar trophoblast happens first, and it will just start invading the uterine wall like this, and this erosion happens from the stratum compactum, because stratum, this is the endometrial lining which is facing the uterine cavity. So, from this to roughly this region you call it a stratum compactum. After that you get the stratum spongiosum. So, the polar trophoblast will first touch the endometrial lining, then it will start invading the endometrium. So, first it will erode the stratum compactum, later it will erode the stratum spongiosum. So, what helps in the erosion of stratum compactum and stratum spongiosum? It is with the help of an enzyme known as trypsin. So, it is just like trypsin like enzyme, the proteolytic action of the trypsin-like enzymes, and which is secreting this trypsin-like enzymes? It is again secreted by the trophoblast. So, the trophoblast, the polar trophoblast, as and when it touches the endometrial lining, it will start liberating a proteolytic enzyme, that is trypsin-like enzyme, and it will start eroding the layers of the endometrium. First it will start eroding the stratum compactum and it will start entering into the endometrial wall. Later it will invade the stratum spongiosum. The embedding process or the implantation process usually starts by 6th day or 7th day after fertilization, and it is not just a one-day process. It will start the process on 6th day or 7th day and it will continue the process until say 12th day of after fertilization. So, this is how the embedding process starts, and to which part of the uterine wall you get this implantation done. So, the implantation site, the common implantation site is the junction of the fundus and the posterior wall of the body of uterus, or roughly we can say that the implantation happens in the upper segment of the uterus. So, if the implantation happens anywhere in the upper segment, whether it be the anterior wall or whether it be the posterior wall, if it is in the upper segment of the uterine cavity, you call it as normal implantation. So, if we summarize, the chief events which occur after fertilization are, it will undergo repeated mitotic division, that is known as cleavage division. As a result, there is formation of morula. As the uterine milk enters the morula, there will be a cavity formed. That cavity is known as blastocoele. So, this morula with the blastocoele will result in the formation of blastocyst. Up to this stage, starting from cleavage division, and it is again after the formation of morula and after the formation of blastocyst, even up to this stage you have a covering for all these, that is known as zona pellucida, and it is the zona pellucida which is actually preventing the premature implantation of the developing zygote. So, after the formation of blastocyst, it is time for the zygote to get implanted. So, at this moment the zona pellucida will be removed and the blastocyst will be implanted into the uterine cavity. And the normal site of implantation is usually in the upper segment of the uterus. This can be considered as the upper segment and this can be considered as the lower segment. So, anywhere in the upper segment of the uterus, you call it as normal implantation, and this is made possible by the removal of zona pellucida. So, is there any chance for the zygote to get abnormally implanted? Yes, it can also happen. So, if the implantation occurs in abnormal sites, you call it as ectopic pregnancy. So, anywhere other than the normal expected implantation site, you call it as ectopic pregnancy. So, the ectopic pregnancy can happen either within the uterus or outside the uterus. How can an ectopic pregnancy happen within the uterus? Because we said the normal implantation, for the normal implantation to take place, it should get implanted in the upper segment. So, if the implantation is happening in the lower segment, we can call it as intrauterine ectopic pregnancy, because the pregnancy is still happening within the uterus but not at a normal site. So, we have to call it as ectopic but within the uterine cavity. So, it is a case of intrauterine ectopic pregnancy. And what do you mean by extrauterine ectopic pregnancy? Extrauterine means anywhere outside the uterus, if it is getting implanted, you call it as extrauterine ectopic pregnancy. So, what are the options we have for the extrauterine pregnancy? One is, we can have the implantation occurring in the ovary, because the fertilized ovum will be expelled through the uterine tube in a reverse direction and it will reach the ovary. So, what will happen? We have the implantation in the ovary. This condition is known as primary ovarian pregnancy. What are the other options? If it is not reaching the ovary, it will be just floating in the abdominal cavity, or it may reach the pouch of Douglas, or it may go to the greater omentum. So, in that case, the implantation is happening in the abdominal cavity. What are the other options? We have the abnormal implantation happening anywhere along the uterine tube, because if it is getting implanted in the uterine tube, again we call it as ectopic pregnancy. So, that condition is known as tubal pregnancy, and that is a very common condition in as far as abnormal implantation is concerned. So, abnormal implantation, the commonest condition is an abnormal implantation in the uterine tube. So, that condition is known as tubal pregnancy. So, where all positions or at which all positions you will get abnormal implantation along the uterine tube. So, it can happen in the infundibular region, you can get the implantation in the interstitial portion, the ampullary region, the isthmus portion, everywhere you can get the abnormal implantation of the zygote. Now, intrauterine abnormal implantation we have already mentioned. If it is not getting implanted in the upper segment of the uterus, if it is getting implanted in the lower segment, you call it as intrauterine abnormal implantation. And this condition where you get this abnormal implantation in the lower segment, this condition is known as placenta previa. So, that was the slide with which we started this session. So, let us see what is placenta previa. So, placenta previa is an example for abnormal implantation, and abnormal implantation here happens in the lower uterine segment. So, when we say or when we grade placenta previa, there are about 4 grades for the placenta previa. The first one is grade 1, grade 1 placenta previa. So, in all the 4 grades of placenta previa, the placenta will be in the lower uterine segment. That point you have to keep in mind. Let us see what is happening in the grade 1 placenta previa. Here the placenta is actually lying in the lower segment, that is it. It is not going towards the os, it is just getting implanted in the lower uterine segment. What do you mean by grade 2? The placenta, again it is in the lower uterine segment, but it is more closer to the internal os. So, that is grade 2. Which is the next grade? That is known as grade 3 placenta previa. What happens in grade 3? We can see that again the placenta is in the lower uterine segment, but it is reaching the internal os, it is covering the internal os, but it is said that if we dilate the internal os, then the placenta will not be covering the internal os, it will just move apart. If we dilate the internal os, the placenta will just move apart, then it will not be occluding the internal os. That is grade 3 placenta previa. And what is this condition? This condition is known as grade 4 placenta previa. In this case, the placenta we can see that the placenta is just overlying the internal os. It just completely covers the internal os. Even if you dilate the os, it will be still covering the internal os. That condition is known as grade 4 placenta previa. So, these are the 4 grades of placenta previa. Grade 1, the placenta lies in the lower uterine segment, but it is not reaching to the internal os. Grade 2, placenta again lies in the lower uterine segment, but it is reaching up to the internal os. Grade 3, the placenta is covering the internal os, but it is said that if you dilate the internal os, it will move away from the internal os. And grade 4, the placenta completely covers the internal os, and even if you dilate, you will not be able to move the placenta. So, actually the grade 4 placenta previa is very dangerous, and all these cases might present with preterm labor and painless bleeding during the late stages of pregnancy. So, that is all about implantation in a nutshell. Thank you.